Let's move on to chapter 7 on page 95 and 96. We have been discussing about mitochondria, endoplasmic reticulum, like different organelles which are present in the cytoplasm of the cell. Which is cytoplasm? It is a place in which liquids are present, fluids are present between cell membrane and nuclear membrane, that is nucleus. In this cytoplasm, many organelles are embedded. Mitochondria is one of the organelles which is situated in the cytoplasm of a cell. This mitochondria is also called powerhouse of the cell because it produces ATP molecules, which is a source of energy. Every component of the cell uses this energy in the form of ATP, which is produced by mitochondria organelles. Next, we studied endoplasmic reticulum. There are two types of endoplasmic reticulum, that is smooth endoplasmic reticulum and rough endoplasmic reticulum. Why this is called rough endoplasmic reticulum? Because on its surface ribosome is attached to it. That is what it looks like, rough surface. It is called as rough endoplasmic reticulum. This endoplasmic reticulum is a site of protein and lipid synthesis. That means this organelle produces protein and lipid molecules which are useful for formation of cell membrane. And also we have studied about Golgi complex. This Golgi complex is also called as Golgi apparatus and Golgi bodies. The materials or components which are produced from ER is packed and dispersed and distributed to various target sites in and out of the cell through this specific organelle Golgi apparatus. Golgi apparatus can be called as Golgi complex. It also secretes some enzymes, proteins as well as hormones. The next organelle is lysosome, which is called a suicidal bag of a cell. This lysosome is situated in the cytoplasm of the cell, and in emergency situation or in any damage happened to the cell, lysosome enzymes which digest that own cell, it digests its own cell, as well as if any damage happened to the organelles, any other organelles, this lysosome digests that organelles also. So it is called as suicidal bug. So in emergency situation, they burst and the enzymes digest itself. Itself means digest its own cell. But plant cells are exceptional to this organelles. Lysosome is absent in plant cell. Part of the cell is ribosome. It is a site of protein synthesis. This ribosome is produced by nucleolus. That means it is produced in nucleolus, which is present in the nucleus of the cell. The next organelle is vacuole, which is present in the cytoplasm, which stores amount of water, large amount of water, and food materials, waste material, excess material. These and all stored in the sap of the vacuole. What is sap? Sap is a kind of fluid which is present inside the nucleus, which consists of these all things like food waste materials, inorganic salts, etc. Generally, plant cells are having large sized vacuole which occupies around 50 to 90% of the cell volume, whereas animal cell has small sized vacuole. The next topic is differences between plant cell and animal cell. Plant cells are large in size than animal cell. In plant cell, cell wall is present, whereas in animal cell, cell wall is absent. Plastids are present in plant cell and plastids are absent in animal cell. The next difference is vacuoles are large size in plant cell, whereas in animal cell, vacuoles are small in size as well as many number. And centrosome is absent in plant cell. Centrosome is present in animal cell. Lysosome is present in animal cell, but lysosome is absent in plant cell. Plant tissue includes meristematic tissue, which help for plant growth, and dermal tissue, which is the covering of the plant. This dermal tissue is a protective layer of the plant cell called epidermis. The vascular tissue includes xylem and phloem, which conduct water and food materials. And the ground tissue of the plant includes sclerenchyma, collenchyma, and parenchyma, which is the site for photosynthesis to occur, as well as which provide these tissues provide mechanical strength to the plant. Activity 6 is to show the vascular tissue or vascular bundles like xylem and phloem cells, which is present in the stem. Materials record are permanent slide of TS. What is TS? It is a transverse section of a stem and also microscope. Procedure: take the slide of this transverse section of the stem, that means cut portion of the stem, cross cut portion of the stem, and put it under the microscope. We can find the tissues like xylem and phloem, and we can see clearly how they are arranged in bundles. These bundles are called vascular bundles. Animals also having various tissues like epithelial tissue, nerve tissue, muscular tissue, connective tissue, etc. Here, blood is a transporter of our body. Blood carries waste materials and fine food materials, nutrients and water particles. So, blood is called the vascular tissue for us. Blood is a fluid vascular tissue which contains blood cell like RBC, red blood cell, white blood cell, platelets. Now let's move on to activity 7 is to observe the permanent slide of blood smear. Blood smear is nothing but it is a kind of blood test, blood strain on the blood layer or blood film on the slide. With this, we are going to test the components, or we are going to see or observe the components in the blood. Here materials required are microscope, permanent slide of blood smear. We keep this slide, blood smear slide, under the microscope. We can observe that 3 different kinds of cells in the blood. That is red blood cell. That is the other name of red blood cell is erythrocyte, and white blood cell, the other name is leukocyte, and platelets, the other name is thrombocyte. These 3 different cells we can see in the blood. These are the important components of the blood. We all grown up from single organism. Every cell exists from pre-existing cell. That means zygote is a small organism, small cell from which we all are developed. What does it mean? Every single organism divides and multiplies and form a big organism. Every small cell, every single cell undergo division and form multiple cell and develop big organism. This is called cell division. We human being develop from single or cell, that is zygote. It undergoes division and form a complete organ or organism. You look at us, we such a living organism are made up of billion, trillion of cells. This trillion of cells develop from one single zygote. So single cell produces billions of cells. This process of division of one cell into two into more, this is called cell division. So from the parent cell, daughter cell is formed by cell division. Daughter cells resemble the parent cell in a number of characters. Many characters of daughter cell is similar to parent cell. Daughter cell grow into maturity and it go for division again. That matured cell again go for division, cell division. Such a cell division is required for our body because it replaces the worn out tissue or worn out cells or damaged cells. Skin cell continue to divide throughout our life, but nerve cell do not divide after a certain age. The next topic is prokaryotic and eukaryotic cell. These overall cells are divided into 2 groups, that is prokaryotic and eukaryotic. What is the difference then? Prokaryotic cells are much different from eukaryotic cells. In what way they are different from each other? The nucleus in prokaryotic cells are not well organized. That is, in eukaryotic cells, that nucleus is surrounded by nuclear membrane. But in prokaryotic cell, there is no nuclear membrane. These chromosomes are spread everywhere. But in eukaryotic cells, having well organized nucleus and nuclear membrane and organelles. Pro means primitive, karyon means nucleus. That prokaryotic means primitive nucleus, that means not well organized nucleus. These cells are bacteria and blue-green algae. These are having not well developed nucleus. Eukaryotics are plants, animals, human beings, etc. And also in prokaryotic cell, this endoplasmic reticulum, Golgi complex, lysosome, these all organelles are absent. But in eukaryotic, these all organelles are present. These are the vast difference between prokaryotic and eukaryotic. So we all are eukaryotic cells or eukaryotic organisms.